Thanks. Thanks, Andrea, for the presentation, and thanks to the organizing committee to give me the possibility to present you our efforts in developing new antibody drug conjugates for cancer and beyond. Antibody drug conjugates, as many speakers shows this morning, are composed by a monoclonal antibody specific for antigen, hopefully selectively expressed in cancer cells, and connected by a properly designed linker to a cytotoxic molecule here called payload. When the ADC arrives in tumor cells, it's able to bind its specific antigens, and usually the mechanism of action is an internalization of the ADCs by endocytosis, and the endosome then mature into a lysosome. Here, inside the lysosome, the ADC can find different pH conditions and enzymes that are able, depending on how we design this linker, to release the drug inside the tumor cell and just producing cell death. There are available even antibodies that are not internalized and that can be used to produce ADCs. Example from Philogen and Philochem are reported in the literature. The difference is just in the linker, on how you design the linkers to get the drug be released outside the cell. Here there are the ADCs actually approved in the market. They are 14, and they start to be approved in 2011, the first one, and actually there are more than 100 ADCs in clinical trials. Why companies are actually so interested in ADCs? We start to work on this field in 2014, so we were almost here, just 2 ADCs were approved, and the idea was that if you have a molecule that is already in the market for the treatment of any disease, and if you have a monoclonal antibody which is actually applied in immunotherapy, you have just to connect them together, and you can skip most of the clinical trial phases, so you can arrive in the market quite easily. Actually, the situation is a little bit different, because going on with the discoveries of new ADCs and with the technology, it's much more mature, so there are many requirements reported, but generally it's a kind of Lego approach, and it's even easy to have something that you can patent, because you have just to change the monoclonal antibody or the payload or the linker, and you have something new. If you have a linker payload system and you change the monoclonal antibody, you can have something which is active against different targets, and that you can use in different diseases. And you can collect a library of different ADCs for different applications, and in this game there is a lot to do, working both in new payloads, new linkers, new monoclonal antibodies, and even in new way to connect the linker payload system to the monoclonal antibody. Any part of this, it's playing a crucial role. Our idea is that this approach will be interesting in the future in precision medicine, because we can think to have a patient coming in the hospital and you have the diagnosis of a cancer, so you can go on with the analysis and you can establish that his or her tumor cells express a specific antigen. So you can try to synthesize a monoclonal antibody specific for this antigen. In this assay, you can find small molecules active on these cancer strains, and you can put them together and treat the cancer of Mary or whatever, and have hopefully a complete recovery. I say a complete recovery because in our experience, what we did with the ADCs we develop is that we have a complete eradication of cancer. We work on melanoma, in non-small lung cancer, in pancreatic cancer strains in different way. With some of our molecules, we are actually in monkeys. And what the biologists observe, I'm an organic chemist, so this is not my part, but is that we have a complete eradication of the cancer. And working on this part, in the chemical part of ADC development, what I realized is that, okay, it's a Lego approach, but working on that, it's much more similar to a jigsaw puzzle, in which do you not really need only to have the right pieces, but you even need to put them together in the right sequence. Otherwise, you are not able to have your final compound, because it's quite common in our lab, I don't know in the others, to work for months in the synthesis of linker payload system, and then arriving at the bioconjugation step, we are not able to connect the monoclonal antibody with this system. So you have to synthesize a new linker or to change the amino acid residues of the monoclonal antibody you want to use, or you have to pre-functionalize the monoclonal antibody, but in any case you lose a lot of time. So that's why in my research, in the context of the Tuscany Health Ecosystem project in Spoke 6, I start on study and developing a machine learning model for the prediction of bioconjugation. And we have been able, with Lorenzo in our group, starting from the data that we have in our lab, because working on this field since 2014, we have a lot of failure in bioconjugation, several success, but we have data that were homogeneous, and this is crucial to have a good machine learning model. And our system, it's able actually to predict if a linker payload system can be bioconjugated or not with your monoclonal antibody, and eventually how many molecules of your linker payload system in certain experimental conditions can be charged over the monoclonal antibody. This is a crucial point, because the drug to antibody ratio, that means the number of molecules that are charged over the monoclonal antibody, it's a very important parameter to obtain an effective ADC. And it needs to be in between 2 and 8 to avoid a loss of efficacy of the monoclonal antibody in recognize the antigen or aggregation processes. So at the moment, this is under reviewing process, and we obtain good results in prediction. Any part of the ADC, it's important. So another part of our research, it's focused on development of different linkers. Here I just reported a simple system, but we developed several over the year, and we are still working on this part. And we work even in finding new molecules, new functional groups that can be used for the bioconjugation with different amino acids over the monoclonal antibody. Even this part, it's important, because these two together are the ones that are important to get for the bioconjugation, an homogeneous ADC, and on the other hand, a linker that needs to be stable in the plasma, but need to be able to release your drug once the ADC is internalized. Then we move to other applications than cancer by changing the monoclonal antibodies used, and particularly in collaboration with Tuscany Life Sciences and the group of Claudia Sala, we work on anti-COVID ADCs, as well in aptamers drug conjugates and in nanobodies drug conjugates in collaboration with the Institut Pasteur, again for application in virus. But this is possible just because several years ago, when we started, we have been able to develop, to demonstrate that it's not mandatory to have a cytotoxic molecule to have an active ADC. This is a crucial point, because when we start, and even now, if you look at the ADCs approved in the market, I reported in the same colors the ADCs that are composed by the same payload, that means the same cytotoxic drug and just a different monoclonal antibody. And except for the green one that have topoisomerase 1 inhibitor as a payload, all the others are charged with natural products which are very, very cytotoxic. The EC50, it's in the range of in the picomolar range, and the idea at the beginning was that if you have a monoclonal antibody and you have just 2, 3, or 4 molecules that are connected on it, to have something which is active in tumor cells, you need something which is very, very cytotoxic. And this was the first choice. And when we start, the idea was that it was not possible to use other type of molecules to have this activity. And using cytotoxic molecule, it's somehow limiting the application of ADCs to cancer. And on the other hand, if by chance you have the release of the drug in the plasma or in other tissues, you have something which is really cytotoxic, and you have a high risk. So the question was, in collaboration with Alpha Wasserman, if it was really mandatory to have these kind of molecules charged over a monoclonal antibody to have an effective ADCs. And the idea, as the company had developed in collaboration with our group an inhibitor of histone deacetylase, was to investigate if it was possible to have an epigenetic modulation by using ADCs. And it was possible. We have been happy for this. Since we discovered and we developed this part by using vorinostat and then all the HDAC inhibitors reported in the literature, again with still with Alpha Wasserman, we realized that the hardest part when you are using compounds that are not very cytotoxic, it's represented by the biological assay you have to do. Because at the beginning, we have no data in vitro assays for any of our compounds. And this is the demonstration on how a collaboration with a company can be an added value in this case, because if it was an academic project, we probably stop saying, okay, it's not possible to do that. But the company was convinced that we can get the activity, and so we directly move to the mouse model. And in vivo studies, the products, some of the products were really, really active. Then we went back and we set up a system for the in vitro assays. But in vitro assays, usually for the first time, you do them in different models, completely fail. So we learn something from this. And then we move to different payloads, such as MR11 inhibitors. We developed some years ago. These ones are good for to be used as radiosensitizers, so we are bioconjugating them. And then we have small inhibitors, that means modulators of the EGOG signaling pathway. And we did this even thanks to the NRRP project, and as well as proteids. So the ability, the demonstration that it's possible to use even molecules that are not cytotoxic open to new projects. Here they are reported, some we are working on antiviral therapy, antitubercular one, as well in the use with using our linkers, especially our payloads, to use different biomolecules as carrier for our compounds, in particularly aptamers for the glioblastoma, and ferritin inside the context of spoke number 6 with Professor Castagna in the University of Siena for the treatment of anaplastic carcinoma of thyroides. This is possible thanks to the work of a lot of people. I didn't report the biologists, they are reported in the patent and in the papers. I reported the people in my lab actually working in the project. Juicy is here with us, and basically she's coordinating most of the part. She came back thanks to the project from a company to work in the university, and she brings with her an added value for my lab. So I'm really happy, and I really thank this project even for this part. Giovanni, he's has a grant from the project and is working in the cyclopamine part, while Demetra is working in developing new linker, and Lorenzo in the machine learning approach. Maria, it's more focused on the payloads, and Francesca, it's actually working in phylochem, and she basically works in different parts such as cyclopamine and linker, and I would like to thank her as well. And for the financial support, obviously, I thank the Spoke 6 of the Tuscany Health Ecosystem and the Regione Toscana for these different projects, PRIN 2022 for the part of anti-retub on tuberculosis, PRIN 2020 for the FAITH project on COVID-19, AIRC and Alpha Wasserman and Ricordati for the ADCs projects in cancer. And your all for kind attention.
Thank you very much, Elena. Thank you so much. Any questions from the audience for Elena?
Thank you very much for your talk. I had a question on how much, if it is a problem when in an ADC complex, the escape from the endocytic compartment where the complex is internalized to progress onto the desired target, so to escape the endocytic bicycle.
You mean the bystander effects?
No, how precise you can be in once the payload is internalized.
Okay, so once the payload is internalized, usually you are quite precise. The bystander effect, the so-called bystander effect, it's quite known. So in some cases, what happens is that the molecules, once released inside the cells, it's moving outside the cells and in the surroundings, and it can act in the cell that surround the target cell. This is not usually considered as a problem, but it's considered as an added value, because you can in this way target molecules that do not express the antigen and are still tumor cells. It depends on how this part is important and how many of these molecules are able to pass the membrane. But in general, there are even some softwares and systems developed over the years that are able to predict, depending on the molecule, on your payload, how many of these molecules will move outside the cells and enter in the environment. Thank you.